Hi, my name is Caitlin Murphy. My presentation is on stem cell culture for Module 5. Stem cell culture is a laboratory technique used to grow and maintain stem cells in controlled conditions outside their natural environment. They are undifferentiated cells that have the potential to basically be changed into various specialized cell types in the body. The technique of stem cell culture involves creating an environment that mimics necessary conditions for stem cell growth and self-renewal. This process typically begins with the isolation of stem cells, which can come from embryonic tissue, adult tissue, or induced pluripotent stem cells, which come from reprogrammed adult cells. The isolated stem cells are then placed in a culture dish in order to grow and proliferate. The culture dish is usually coated with a substrate that promotes stem cell attachment and growth, and they are typically grown as colonies or clusters in the dish, where they are regularly monitored to ensure optimal conditions for their growth, meaning that they have enough of the necessary ingredients, culture medium, to continue growing. How does this technique tie into the module? This technique of stem cell culture is essential for studying the biology of stem cells and investigating their differentiation potential and developing therapies based on stem cell and transplantation. Stem cell culture ties into this lecture because it follows similar principles and practices used for the culture of other eukaryotic cell types. It uses isolation, expansion, differentiation, and characterization. This image, it is showing the stem cell aggregate formation. In image A, it shows stem cells that form small aggregates or large agglomerates suspension. In B, it shows the transport parameters of the system. And in C, it shows the collision frequency, which is modulated by the mass and number of cells in the aggregates in suspension. And then in D, it shows the binding kinetics of the adhesion molecules, such as integrins and cadherins. So the limitations of stem cell culture are maintaining pluripotency or multipotency of stem cells over extended periods of time. So differentiation can occur spontaneously in culture, leading to the loss of desired stem cell properties. Contamination can also occur, which can compromise the purity and integrity of the stem cell culture, and the genetic stability of the stem cells may be affected during long-term culture, which can lead to genomic alterations. But in this image, it shows the handicaps of EV research, which are the production of sufficient material in a consistent and effective way, controlling stem cell growth conditions with physical properties, which are the cell culture system and cell density, oxygen tension and media preconditioning with soluble factors, which can influence paracrine activity by modulating EVs. Advantages of this technique versus other techniques. So compared to studying stem cells in vivo, these stem cell culture systems provide a controlled and reproducible environment, allowing for more accurate experimental manipulation. And it also allows the expansion of stem cell populations, providing larger quantities of cells for downstream applications. They are also good for the generation of standardized cell lines, which can be shared among researchers and used as models for various studies. My image 3, this is showing a three-dimensional growth of GFP-labeled mouse ESCs and prefabricated PDMS scaffolds. So in image A, showing the schematic of ESCs seeding in prefabricated PDM, and B, it shows macroscopic images of the PDM scaffolds. And C through E represent confocal images of green fluorescent labeled ESCs grown in prefabricated PDMS scaffolds at different numbers of days. Okay, so the controls that are in stem cell culture are negative controls. These are basically, there's nothing in them, so it allows for kind of a baseline of what the stem cells should look like if basically they don't grow or there's nothing else there. There are positive controls, which involve culturing the stem cells under conditions known to induce specific responses, which can help to validate the functionality of the stem cells and the experimental setup. Then there are isotype controls, which involve the use of antibodies for cell surface marker characterization. And they are antibodies with the same isotype as the primary antibodies used in the experiment, but they do not recognize the target antigens. Here are my citations. Thank you.